Hey, Mr. Wartsky here again, and this is the beginning of the fourth screencast in the organic molecule series. Some of them have been quite long. This should be the last slide that you saw in the third screencast. So we're going to continue to talk about shapes and talk about the types of proteins. So if we change the shape of a protein, that's called denaturing the protein, or we denature, the protein denatures. So how can you denature a protein? Basically, you break hydrogen bonds. So how do you break hydrogen bonds? First, you decrease the pH. You introduce hydrogen ions. So hydrogen ions will bind to the partially negative atom, and the hydrogen bonds can't form. If you increase the heat, you move the hydrogens really fast. The hydrogen bonds break and reform, and as they break and reform, they reform in a different place. That changes the shape. If you add free radicals, the free radicals are OH groups with an extra electron. They are attracted to the partially positive hydrogen, and if they bind to the partially positive hydrogen, hydrogen bonds will break and they can't form. So that's how you denature, and hopefully you got all that down. Again, if you didn't get it down, pause it, go back, and listen to it. Hopefully it's not that bad. So we're going to talk about the different types of proteins. Binding proteins or transport proteins bind and carry things from place to place, like hemoglobin carries oxygen from place to place. Structural proteins provide support and shape. For example, collagen makes up about 25% of your body mass. It's your connective tissue. Connective tissue holds you together. Elastin is another type of connective tissue which holds you together, provides structure. And then keratin makes up your hair, your nails, your feathers, your claws, your horns, your beaks. That's found in keratin. By the way, you cannot digest keratin, so please don't try to eat your fingernails or feathers or hair. That's a big one. You can't digest hair. Hormones are other types of hormones. Other types of proteins. We have peptide hormones, like growth hormone. And these things coordinate your body's activities as well as maintains homeostasis, which is important. Other types of proteins are receptor proteins. They're on the cell membrane and they receive molecules. Those molecules are called ligands. That's a general term for any molecule that binds to a receptor. Defense, we have antibodies. Proteins bind to foreign molecules or pathogens that hold them so that our cells can eat them. And then the one that we're going to spend a lot of time in are enzymes. And enzymes decrease the activation energy of a reaction. So here we have the amount of energy, and the activation energy is the amount of energy needed for a reaction to start. So we usually have to increase heat, right? We provide heat. What happens is the molecules move really fast. They bump into each other, and as they bump into each other, they react. If you're going to increase the reaction rate, you have to keep on increasing the temperature. But the problem is, if you keep on increasing the temperature inside of us, our biological systems, our hydrogen bonds and proteins are going to break, our proteins will denature, and we will die. So we can't have that happen. So how do we increase temperature and at the same time keep our proteins good? How do we have reactions without denaturing our proteins? So the answer is enzymes. So in this little graph, it shows you that without enzymes, we have a high activation energy. But with enzymes, the amount of energy needed is low. And that low temperature right there, the low amount of energy, allows us to live. So that's how you read that graph. You might want to include that graph in your notes. So enzymes have a specific shape, and we know that those specific shapes are determined by the 4 levels of protein conformation: primary, secondary, tertiary, and quaternary. We got that. The enzymes have what is known as an active site, which is a groove or depression on the enzyme, which allows substrates to enter. Now, substrates are molecules that start a reaction, and products are molecules that end a reaction. So substrates will enter and bind to the active site. Now, the shape of the active site is extremely important because that will determine what type of substrates will enter and react. So the reaction or the enzyme function is determined by the shape of the active site, and that determines the type of substrates that can react. So enzymes provide a place for reactions to occur. Specific shapes allow specific substrates to enter. They are known as catalysts. Now you need to write this down because a catalyst will increase the rate of reaction by decreasing the activation energy without becoming part of the reaction. Again, sure. Catalysts will increase the rate of reaction by decreasing the amount of active energy needed, amount of activation energy needed, and without becoming part of the reaction. Mess that up. So the old idea was that it was a lock and key. So the key was the substrate, the lock was the enzyme, and it had to be a specific shape to fit into the enzyme and to unlock the lock, right? Now, the induced fit model means that the enzyme is a little bit more flexible. It's not as rigid as once thought. So it can accept similar looking substrates into the active site. So the enzyme substrate complex, enzyme product complex, and enzyme product, all terms that we need for an enzyme reaction, which is on the next slide. I would like you to draw this because this is relatively simple. We have the substrate up here. Notice it has a shape. We have the active site here, which also has a shape. This is the enzyme plus substrate. The substrate enters the active site, so this is the enzyme substrate complex. The substrate's reactive form product, which forms the enzyme product complex. And then the products leave, forming the enzyme plus products. Now, what type of reaction we know of two is either synthesis or hydrolysis. What reaction do we have here? And if you said synthesis, you would be wrong. It is a hydrolysis reaction because we are taking a large molecule and we're breaking up into smaller products. Large substrate, smaller products, hydrolysis. So you're asking, well, what about synthesis? I want to draw a synthesis. Please, can I draw a synthesis? And sure you can. So we have substrate 1, substrate 2, enzyme. Here are the active sites. This is the enzyme plus substrates. We form an enzyme substrate complex where the substrates enter. The substrates form a product, which is the enzyme product complex, and then the product leaves. So we took two smaller substrates, formed a larger product. This is, yes, you got it right this time, synthesis. Very good. Yes, I would like you to get both hydrolysis and synthesis, and we'll do something with this in class. So what affects the rate of enzyme reaction? Great question. Temperature. So we increase the temperature initially, the molecules move faster, they bump into each other, so the reaction rate will increase. But then it gets to about 40 degrees Celsius and the hydrogen bonds break. And the hydrogen bonds break, the enzymes fall apart or denature and change shape. Reactions will stop. Time. Initially, the substrate concentration is high. The reaction rate is fast because a lot of substrates can enter the enzymes and we have a lot of product formed. But over time, the amount of product increases. So the product concentration increases, the number of substrates decrease, so the reaction rate will actually slow down. Sometimes the product enters the active site and prevents the substrates from entering, and that's called negative feedback. This is important. We'll have an activity that deals with this. If you decrease the pH, you will denature the enzyme. If you change the shape of the enzyme, reaction stops. Hydroxyl or free radicals, you increase them, you will denature the enzyme because increased free radicals will denature proteins. Reaction will stop. Now, this is a hydroxyl radical right here. It's usually produced by your production of energy. We have an enzyme called superoxide dismutase, which will take your free radicals and form hydrogen peroxide. The hydrogen peroxide is broken down by another enzyme called catalase, and you're good. Now, antioxidants will protect you from free radicals. So that's why antioxidants are good. Because if you have free radicals, you'll be denaturing your proteins everywhere, and that is bad. Cofactors and coenzymes are found in active sites. They provide shape. That's what your vitamins and minerals do. And if you have the right shape, you have the right reaction. But if you don't have your cofactors and coenzymes, your vitamins and minerals, you won't have the right shape. You won't be able to accept the substrate, and you will have problems. Don't like problems. Reactions won't happen. There's also another way to control enzyme reactions is through allosteric inhibition. Sometimes on some enzymes, there's a secondary site, which is called the allosteric site. And if I can find the mouse right here is the allosteric site here, and right there's the allosteric site there. So a molecule can bind to the allosteric site. It will change the shape of the active site, and one of two things can happen. Either this side says the reaction stops, or sometimes, very rarely, it can change the shape so that it can accept substrates. But usually these are part of multi-step reactions, and they stop the reaction. So usually it's this one here. You need to draw the picture, by all means, draw the picture. And we're going to stop there for today. I hope this helped.